Hello, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by BioAgilytics. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. This program is being recorded, and the video will be available on AAPS Science 360. It is now my pleasure to introduce today's moderator, Jessica Bridges-Weaver. Jessica Bridges-Weaver is the scientific officer at BioAgilytics, and she brings over 20 years of expertise in pharmaceutical development of upstream and downstream processes in analytical testing in support of chemistry, manufacturing, and controls. Jessica, the floor is yours.
Thank you, Stacy. Welcome everyone to today's eChalk Talk. We're going to cover control strategies around ADCs and monoclonal antibodies. And we're going to hear from some subject matter experts, Brian and Daniel, and I'll introduce them shortly. First of all, I will just let you know that the slide deck can be made available upon request. So if you would like a copy of the slide deck, please let us know. Today, we're going to cover a few objectives of today's presentation, an overview of monoclonal antibodies and ADCs. Brian's going to speak specifically towards LCMS characterization and SCC, CE, and IC-IEF characterization. And then Daniel's going to follow up with some SPR. After that, we'll follow up with Q&A. So just a few objectives for today. So we're going to explore case studies demonstrating the application of, as I mentioned, LCMS, some size exclusion, capillary electrophoresis, as well as SPR, and some complementary techniques that we use here at BioAgilytics to characterize ADCs and monoclonal antibodies. In this table, we've highlighted a few of the different attributes we're going to discuss specifically today by those platforms. And then we will continue that conversation. Just a little background, as we all know, monoclonal antibodies are the backbone for antibody-drug conjugates. So most monoclonal antibodies, a lot of the testing that we can do to support those are also carried over into ADCs. And the main primary difference is obviously the drug or attached with the linker to make that monoclonal antibody now an ADC. So we do have comprehensive monoclonal antibody and ADC support at BioAgilytics. Today, we're going to solely focus on the CMC analytical support, and we can support a full CISPQ package for your monoclonal antibody or your ADC. But BioAgilytics as a whole can support additional bioanalytical testing for large molecule and small molecule across all of our sites globally. So if you are interested in any clinical trials as well, please reach out for like PK, ADA, NAB assay testing and free payload testing. With that, we're going to kick off with some LCMS characterization. And I would like to introduce to you Brian Thacker. He has a PhD. He's from the University of California in San Diego. He also got his master's in bioengineering. He's been with BioAgilytics now for, I believe, about 5 years, but he's a scientist too with us. He does have over 10 years total of drug development experience. From early phase startup assay development and cell line development through late phase validation. And his contact information is here on the screen. And with that, I will turn this over to Brian.
Thank you, Jessica. Just to kick off here, some brief notes on regulatory guidance for LCMS assays before we get into the technical content. As indicated by ICH guidelines, early phase efforts in development and for the LCMS assays for ADC characterization, they can be used to interrogate intact mass, subunits, peptide mapping, DAR determination, and a host of other things, which we'll touch on in the technical aspects. Early phase, these may be qualified assays, but need not be validated. However, when you move to later phase efforts, then it is expectation that you'll use fully validated methods. And as we'll mention in the next slides, we'll be looking at the critical quality attributes of drug-antibody ratio, distribution of drug, aggregation of the drug molecule, and other related impurities. Let's go to the next slide, Jessica. Okay, so just to provide some context and orientation for the technical details that will come, just providing an overview of the ADC structure. As Jessica mentioned, it's based on a monoclonal antibody backbone, which in and of itself has its own complexity inherent to large molecule protein products in terms of its glycosylation. And you couple that complexity with the added heterogeneity that comes from adding chemically modified drug product to that, you end up with this complex molecule. And there's several methods that need to be brought forward to fully characterize and understand, control the product. Just to provide like a roadmap here, to demonstrate, there's different subunits of this molecule that we'll be talking about later. So the FAB subunit is, the top of the image there, versus the FC subunits, which are at the bottom. Those will be important later on. And also, we'll talk about how some of these, the complexity of the protein can be reduced by using some simple techniques to break down the protein into its component subunits. So, for instance, this enzyme called IDES can be used to cleave the protein at a very specific location, liberating the FAB portion from the FC portion. In addition, we can use a reductant like DTT to reduce the disulfide bonds and provide further separation into subunits. Let's look at the next slide. Here we're just showing briefly like a workflow that we can use to generate different samples that can be analyzed by LCMS methods. So at the top here, we can analyze intact ADC drug product directly, or it can be digested using the IDES enzyme, as I mentioned, to liberate the FC and the FAB portions, subunits. And then that subsequently can be reduced using DTT, which gives further breakdown into the component subunits. Any of these different preparations can be applied to the LCMS to generate separation by chromatography and then MS mass source spectra. The spectra that come out of these analyses is quite complex and difficult to work with manually, so we use a software package like Biopharma Finder. You provide Biopharma Finder with the amino acid sequence of the protein that you're working with and the expected modifications to that sequence, including the drug molecules and glycans that might be attached to it. And then it deconvolutes the data and annotates it according to the masses that it has detected. And we'll show that information in the upcoming slides. Let's go to the next slide here. Okay, so an example here of the kind of data that we can generate looking at the intact ADC molecule to kind of provide orientation. An image of the molecule that we're looking at is at the top right. You'll see that in subsequent slides. But the method here is using a reverse phase column that's intended for intact protein analysis in the LC portion, and then the mass spectrometer is a high resolution Orbitrap Exploris 240. And as I mentioned, the analysis of the raw data is performed in Biopharma Finder. So if we do analysis of the intact protein, we see a lot of information shown on this graph, and I'll just walk through it slowly so to provide context and orientation. What's being shown in the graph is the deconvoluted data. So each peak there represents a mass that has been detected by the mass spectrometer. And the height of those peaks is the relative intensity of each of those signals within the mass spectrometer. You'll see there's lots of different peaks here. But if you look at the x-axis, they all are clustered around mass range that we'd expect for a monoclonal antibody, which is around 150 kD. In addition, you'll see there's this repeating pattern of triplicate peaks that kind of marches across the graph. Those triplicate peaks, if you look at the very bottom, they can be assigned to different levels of drug conjugation to the ADC, and that's depicted in D = 0 through D = 8 at the bottom of the panel. So what we've detected here are ADC molecules that are carrying anywhere from 0 drug molecules, which is an unconjugated monoclonal antibody, all the way up to as many as 8 drug molecules attached to each ADC molecule. There's lots of other stuff in here. For instance, why are there 3 peaks in each of those clusters? The 3 peaks arise from the glycoforms that are present in the sample. we detect two different glycan structures, and there are two glycans attached to each ADC. So the three possible combinations give you those three peaks that you see in each of those repeating structures. Okay, so all this data taken together, Biopharma Finder can use the amount of drug molecules that's detected here with each species to calculate an average drug antibody ratio. That's shown at the top of the panel there. This calculation gives a DAR of just an average DAR of just over 3. So here we have a lot of information on the DAR, on the distribution of drug molecules, on the glycoforms that are present in the sample. What we don't see here is any information on where within the ADC those different molecules might be found, or different conjugations or modifications. And so to get to that, we look at some of the subunits that were liberated using the IDES digest or DTT reduction. Let's look at the next slide. This slide specifically is showing the kind of data that can be generated after using the IDES enzyme to digest the ADC. So at this point, it's broken into its component subunits with the FAB portion and the FC portion separated. We can separate these chromatographically and then look at the masses that are derived from each of those subunits independently. Again, I'm not showing the raw data here, I'm showing the deconvoluted data. And this is the data shown here is specifically for the FAB subunit of the ADC. What we see here again, the masses that are kind of towards the top of the figure there are FAB subunits that have different levels of drug conjugation. So you'll see on the far left, there's that annotated peak that says D0. So there's FAB portion with no drugs attached to it. And then as you march across, you see increasing levels of drug conjugation again. Looking at a simplified analysis like this, there's lots of masses that are not, compared to the previous slide, not so many masses that are relevant, allows us to pick out additional features that were not apparent before. And something of interest that I'm showing in the bottom of the panel, it's outlined with the dotted lines there, is that we can see masses that would be that pertain or assigned to ADC structures where there's a linker present, but there's no drug payload conjugated to the linker. So these would be process impurities. We can see them very clearly and monitor them using the LCMS analysis and understand how they might be changing from batch to batch or through stability studies. What's not shown here is the data from the FC portion, and just for brevity, it's not shown, but what we see in the FC portion is that there's one glycan structure attached to each FC subunit in kind of the canonical location for glycan attachment. And as you'd expect from the intact mass protein data, we see the two different glycoforms, that A2G0F and the A2G1F in that data. Let's go to the next slide, Jessica. Okay, so we've kind of looked at the ADC from an intact mass and a subunit perspective. We can get greater resolution down to the amino acid level by using a peptide mapping method. And I'm just showing the workflow here very briefly. The approach here is to take the intact ADC, trypsin digest it, so you reduce its complexity down to its component tryptic peptides. And some of those peptides will be carrying the drug conjugate still. Those peptides can be analyzed in the LCMS. And again, in this analysis, we're using MS2 fragmentation, which allows us to see masses down to the amino acid level. And then Biopharma Finder also will allow us to deconvolute that data and map the peptides that have been identified to the overall protein structure. If we go to the next slide, Jessica, we can look at some very simplified results for that kind of analysis. What I'm showing on the left in the table is the number of, it's a high level overview of what an analysis like this might generate. You see the number of peaks, MS peaks that are generated for both the light and heavy chains of the antibody. And the sequence coverage here is 100%, meaning that the MS analysis and Biopharma Finder deconvolution was able to identify peptides that would cover 100% of the expected protein sequence. of the underlying mAb for the ADC. So using that information, we can verify the amino acid sequence of the protein. We can also use this data to verify or ascertain which amino acids are carrying the drug conjugate. And that's facilitated in this analysis by the fact that the drug molecule also fragments during MS2 analysis. And that's shown in the bottom left. That's a drug molecule with its linker. A fragmentation, a characteristic fragment ion that arises from fragmentation is this as the ion with the mass 547.22. I'm showing one example on the left. You see the MS spectra. On the top panel there, it's the predicted spectra or the theoretical spectra given the peptide that's being analyzed. You'll see the experimental data down below. In the red masses there, they match up with the predicted masses, verifying that we're seeing the peptide of interest. But you'll see in the far left that I've circled in red, there's an additional ion there, which matches the fragment ion that's expected for the fragmentation of the conjugated drug. So a signal like that verifies that this peptide was carrying the drug conjugate. Okay, let's go to the next slide. So we can use this kind of data to monitor the ADC product as it's produced from batch to batch. We can also use the data to monitor the quality of the product in stability studies. To illustrate this, we've just done a very aggressive heat treatment of ADC over a 24-hour time period. What we did is we determined the average DAR for each of those samples. So you can see at 70 degrees, we get a strong reduction in DAR for the sample. At 50 degrees, we see a slight reduction at the 24-hour time point. Is an interesting finding and allows us to have samples that we can use to illustrate orthogonal techniques, which I'll touch on in the next few slides. Okay. Next slide, Jessica. Okay, so some key takeaways for the LCMS portion here. We can use the LCMS with high resolution to do intact molecule subunit analysis, peptide mapping of the ADC product. And through those analyses, we can monitor multiple attributes of the ADC, including the drug antibody ratio, the conjugation profile of the drug molecules, the glycosylation of the antibody, and process impurities such as those linkers that didn't have any drug molecules attached to them, and to confirm the sequence of the protein that's underlying the ADC structure. Okay, let's go on to some orthogonal separation methods that we can use to further characterize the ADC product. So I'm just going to touch briefly on some capabilities that we have. First, again, we'll look at some regulatory guidance. As with the LCMS approaches, early phase approaches for these separation chemistries are fit for purpose. They may be qualified assays, but need not be qualified. Later phase, according to ICH guidelines, expected to have fully validated methods to do these kinds of analyses. And again, we'll look at aggregation, fragment, charge variance as we look at separation chemistries. Let's go to the next slide. Okay, so we're going to touch on a little bit here of separation by size. So in the first example here, we're looking at size exclusion chromatography. The methods shown there on the left with the size exclusion column, mobile phase, and flow rate that's used. The detection here is UV detection, so we should be picking up all the protein products that come through the column. I'm showing in each of these next few slides representative data from protein products, in this case ADC, that have been held at 4 degrees Celsius. So it's representing the nascent molecule and versus those that have been stressed at 50 degrees Celsius. So we can illustrate how these methods can be used to detect changes in the chemical structures. So here's size exclusion chromatography. As you probably know, molecules are separated by size. with the larger molecules eluting earlier from the column, and then smaller molecules are eluted later. What's shown here is a zoom in of some representative data of ADC. The major ADC peak is quite large there. It goes off the graph. We see a small hump to the left. It's a larger molecular weight product in the 4-degree sample. But then when we look at the 50-degree stress sample, we see the addition of this extra peak, indicating that there's some higher molecular weight structures that are present in the sample after it's been stressed at 50 degrees C. We can attribute these to aggregation of the sample during heat treatment. But beyond that, there's not much we can ascertain from this kind of analysis. If we needed to know more about the chemical nature of those aggregates, we could move this back into a high-resolution mass spec platform using size exclusion to really understand the chemical nature of that higher molecular weight peak. Let's go on to the next slide to illustrate another type of technique we can use. In this case, this is capillary gel electrophoresis. So it's another sizing method, in this case using electrophoresis rather than flow against a matrix to separate things by mass. This is a non-reducing, an example of a non-reducing approach. So we get the proteins separated by size. Again, the main ADC peak is the major ones that you see there that's going off the graph. But we do see differences in the surrounding peaks, indicating that there's some shift in the size of the materials that are being observed in response to the high heat stress treatment. Both at higher molecular weights and lower molecular weights. Okay, let's go to the next slide, Jessica. Another method that we can use is isoelectric focusing, and that's shown here. So in using this method, we're separating the molecules within a pH gradient, allowing them to separate by their pI. A molecule like a monoclonal antibody, which is shown in this figure here, it has a characteristic fingerprint of a pattern that you'd see in a ICIEF run. That fingerprint can be used to ID the protein from batch to batch, but it can also be used to understand how the protein might be changing under stressed conditions. So here another example is of a nascent protein and the stressed protein in the bottom panel. You'll see that the fingerprint is largely the same between the two examples, but after stressing, we see the accumulation of material within that peak that's just sitting to the left of the major peak. Those are slightly more acidic species of protein, and something about the heat treatment is allowing for accumulation of those acidic proteins, and that can be picked up using this kind of analysis. Okay, next slide. Okay, here we're just another illustration of how isoelectric focusing can be used. In this case, we're not looking at a stress condition. We're comparing the monoclonal antibody to its ADC product after drug conjugation. And you can see here that there's this massive shift in pI after the monoclonal antibody has been decorated with the drug conjugate. In this case, it's the conjugation is occurring on lysine residues. So the drug conjugation is binding up the basic residues, allowing the protein to become more acidic, which is reflected here in the data. So this could be used, an approach like this could be used to monitor the process chemistry and understand how different conditions might be affecting the conjugation and the pI of the protein. Next slide. Okay, so just to wrap up here about some key takeaways from the separation methods. Methods that we've shown today can be used to monitor the size and the charge of the proteins, and they could be applied to the process development, batch testing, and stability protocols for each drug product.
Thank you, Brian, for that information on LCMS and other characterization from separation techniques. With that, we're going to turn it over to SPR characterization. And let me introduce you to Daniel Worms. He holds a PhD. He is part of our CMC lab in Germany, in Hamburg. He is a principal investigator there, and he oversees not just CMC, but also pharmacokinetic, immunogenicity, and SPR-based analytical techniques. He can work with establishing them, validation, some sample analysis of multiple different drug development programs. And prior to joining us in 2018, he completed his doctorate in biophysics and structural biology at HHU in Dusseldorf, as well as some research center, FZJ. And his contact information is here for future reach out if you need to speak about SPR. With that, I'm going to turn it over to Daniel.
Thank you, Jessica, for the nice introduction. And we can move on to the next slide, please. Yeah, as in the previous sections, I would like to give you a brief overview of the regulatory guidance that is applicable for SPR assays. So in the early phase, fit for purpose methods can be used to understand the mode of action, binding specificity, relative potency indicators, and a full validation is not required at that stage. In the later phase, SPR methods can be used for release, stability, and comparability testing. And at that stage, they are expected to be fully validated according to the relevant guidances. The critical quality attributes are binding kinetics, receptor interactions, Fc effector function, target engagement, and epitope conservation. Next slide, please. All right. Before we go into some experimental details, I would like to give you a brief introduction into the technical background on SPR, or surface plasmon resonance. In full, we are using a BioCore devices for SPR analysis. And the system consists of a sensor chip that has a gold film, and on top functionalized external molecules to attach ligands to the surface. There's a flow channel to flow an analyte in solution over the system. And there's a prism underneath the sensor chip and a light source that emits polarized light. And the polarized light is then passing through the prism, and the light is then reflected at the interface of the prism and the gold film. And then it is detected on the other side of the prism. Now, not all of the photons are totally reflected. Part of them at a certain specific angle, the so-called SPR angle. The photons from the polarized light are interacting with the gold atoms, creating a so-called surface plasmon, which is represented by this yellow wave. And this is an electron wave that travels along the surface of the gold atoms. And this resonance, or SPR angle, that is highly dependent on the refractive index in the flow channel. And this refractive index changes when molecules start to bind to the surface, or to the ligand that is attached to the surface. And this this energy conversion from light energy into surface plasmon, that is then observed as a local drop in the intensity on the other side of the prism, on the detector. And this angle shifts, as I mentioned, when molecules bind. This allows a label-free detection of the molecular interactions in real time. The signal, or the change of the angular shift, is expressed in resonance or response units and then plotted over time. And this gives a sensorgram, which is shown on the right picture here. So at the beginning of the run, only running buffer is injected into the system. And since the refractive index is not changing, a stable baseline is observed. And as soon as an analyte sample is injected through the flow channel, it starts to bind to the ligand molecule. And one will see an increase of the response units visualized here by an association curve. Then when the analyte injection is stopped, the molecules start to dissociate from the surface, and this is then observed by a dissociation curve. And finally, a regeneration solution is passed over the flow channel to remove any residual analyte molecule from the surface, so that the baseline afterwards returns to the original state. So next slide, please. All right, this is an example of a kinetic assessment of Erbitux binding to EGFR. Erbitux is a monoclonal antibody that binds to the epidermal growth factor receptor, or in short, EGFR. And Erbitux inhibits growth of cell cancers by binding to EGFR, and it also induces antibody-dependent cellular cytotoxicity. In this experiment, Erbitux was captured as the ligand on the sensor chip surface, and EGFR was injected into the system as the analyte in solution at 5 different concentrations. So in theory, one single concentration is sufficient to determine the kinetic parameters. We use at least five different concentrations covering above and below the expected KD to ensure that the global fit is robust, as the kinetic rate constant parameters that are derived from the curve fit are independent of the concentration and need to describe all the observed sensorgrams. So in that case, the association curve and also the dissociation curve were fitted, and the association rate constant, described here as small ka, and the dissociation rate constant, small kd, they were obtained from a global fit, and the ratio of these two rate constants give the equilibrium dissociation constant, the capital Kd, and in that case, it was 1.3 nanomolar for this interaction, which is considered to be high affinity. So next slide, please. Thanks. So another example I would like to show you is a kinetic assessment of trastuzumab and the ADC Cadcyla binding to the HER2 receptor. So trastuzumab is a monoclonal antibody that also binds to the HER2 receptor, which is found on breast and stomach cancer cells, and also inhibits growth of the tumor cells. And Cadcyla is an ADC, which is based on trastuzumab and then uses the linker SMCC to conjugate the small molecule mTANSine. mTANSine binds to microtubules and thereby induces apoptosis of the cancer cells. So here in this example, we also performed a kinetic assessment of both modalities. And interestingly, the association rate constant Ka is quite similar between the two modalities, though the association rate is a little bit faster for Cadcyla. But more importantly, what we observed is that the dissociation rate constant of trastuzumab is twofold higher compared to Cadcyla, which then results in a different or slightly lower overall binding affinity of 4.7 nanomolar, which is 3 times higher, so to say, than the observed binding affinity for Cadcyla. Next slide, please. So this data was then further processed in a kinetic comparison of the sensorgrams of trastuzumab and Cadcyla. In this example, trastuzumab was used as the reference standard. And this evaluation software calculates the average and the standard deviation from replicate injections at each concentration level. And then a similarity window will be set. In this example, was set at the average plus and minus threefold the standard deviation. And the similarity score is then calculated based on the number of points inside and outside the similarity window. So here we compared both phases with each other, so the association phase and the dissociation phase of the two modalities. And so for Cadcyla, the similarity score is 98% compared to trastuzumab in the association phase, while in the dissociation phase, the similarity score is only 62%. Next slide, please. So related to the previous slide, I would like to show you also a case study of a biosimilarity assessment where 3 different biosimilars were compared to the originator trastuzumab. In that case, we did not test only the main target, HER2, but also Fc receptors like CD64, 16A, and the neonatal Fc receptor, FcRn. So as mentioned before, HER2 is localized on cancer cells and is the main target of trastuzumab. And here we see that the overall affinity of the biosimilars is quite similar to trastuzumab and slightly lower or a higher affinity in that case. CD64 is a receptor localized on macrophages and mediates antibody-dependent cellular phagocytosis. And also here the affinity values are very similar or slightly higher or higher affinity compared to trastuzumab. Then for CD16A, this is a receptor which is localized on natural killer cells, and this mediates antibody-dependent cellular cytotoxicity. And in that case, we did not use the one-to-one binding model. Instead, we used the heterogeneous ligand binding model for the fit of the curves. That is due to the different glycosylation patterns of CD16A. And this model has two distinct KD values. And the primarily relevant affinity value here is KD1. And in this case, or in this case study, the affinity values were compared to trastuzumab, they all showed higher affinity. And then last but not least, we also tested the affinity towards the neonatal FC receptor. This receptor is relevant for transport and recycling of IgGs and is an important kinetic feature that affects the half-life of trastuzumab. So in that case, the affinity was quite similar among the biosimilars, except for biosimilar B, which showed a twofold lower affinity value. Next slide, please. Then finally, I would like to give you a brief overview of the validation parameters that need to be assessed in potency and kinetics affinity. Would like to highlight a little bit the similarities, but also the differences and some parameters that are specific to surface plasmon resonance. So for example, parameters such as precision, specificity, and also robustness. These parameters are applicable to both potency and kinetics and affinity assays. Other parameters like accuracy, range, and linearity will only be assessed in potency assays, as these parameters are not applicable to kinetics and affinity assays. And instead, additional system suitability parameters will be assessed. So apart from the immobilization and capture level that is applicable to both, also the KD and the KD range will be assessed. The so-called Rmax value, which is the theoretical maximum binding, and the chi-square value, which is a measure of closeness of the fit with the experimental data. So what is specific to SPR assays are the robustness parameters here, the baseline stability, which I initially mentioned on the first slide, is that a good regeneration protocol needs to be developed to make sure that the baseline returns to the previous state and does not increase or decrease over time. And also the surface performance is important, as this determines the number of cycles and that can be run on one sensor chip. Next slide, please. All right. With that, I would like to conclude my section of this talk and provide a few key takeaways. So SPR is a very versatile platform that supports all stages from drug development to manufacturing. For example, a kinetics affinity ranking of antibody drug candidates during the drug development stage. But it can also support preclinical and clinical testing, for example, in human ADA, including isotyping, neutralizing antibodies for antagonistic drugs, as well as PK and PD assays. And it supports kinetics and affinity for product characterization and potency assay for drug batch release testing. Thank you very much for listening, and I'm happy to answer your questions.
Thank you, Daniel. And following up with the table that we started with at the beginning, where we highlighted just the attributes we were going to discuss today, I've updated that table to show and highlight the attributes that bioagilitics can support for monoclonals ADCs as well as linker, monoclonal and linker analysis for future reference. With that, I'd like to acknowledge both our US and our EU analytical teams for helping generate the data that was presented today, as well as sponsors who place their trust in us with their data, and also for AAPS for helping us facilitate this webinar series and bringing us all together today. I'd also like to comment that all images and likeness were built in BioRender, and so for the antibodies and the depiction of assays. With that, I'll be turning it over for questions. So Daniel and Brian, if you would come back on camera, we've got a couple questions that for each of you to answer. First off, I'm gonna start with you, Daniel. Resonance time for the duration of injection cycles. What is the runtime for the assays for your SPR?
Yeah, thanks. That's a good question. So typically in kinetic assays, the injection phase of the analytes is roughly 30 to 60 seconds. And then for the dissociation phase, 1 to 3 minutes. That is the typical starting point. But depending on the reaction, it might be the case that the injection and also the dissociation needs to be extended. And typical runtime is depending on the number of analytes and samples that need to be tested from a couple of hours up until 48 hours. That is the maximum the device can do.
All right, thank you for that. And Brian, I've got one for you, asking that since ADCs have antibodies, linkers, drugs, varying DARs, and they can have a large range of different molecular weights. So how do you account for the range when using complex samples? And is the digestion that you mentioned sufficient?
Yeah, in our experience, the digestion is not only sufficient, it's generally necessary to really pick out all the structural features that are in there. We were able to deconvolute the ADC data that I showed pretty easily in this test case. We have other products that we've analyzed that are a little bit more ambiguous, initially at least. For instance, a monoclonal antibody product that we were working with, it had more than just the two kind of canonical glycans. It carried additional glycans that were, it was really difficult to annotate the data and assign the structural features based on intact mass analysis alone. Once we did the subunit analysis, then we started picking apart those structural features and to work our way backwards to understand those structural features like the additional glycans. So yes, the digestion is typically sufficient and also often necessary to understand structure of the protein.
All right, thank you for that, Brian. One more for you that's on the screen is, how do you optimize the LCMS method for intact ADCs versus characterizing release fragments or payloads?
Yeah, so I think this is referring to the intact mass versus the peptide analysis. So the intact mass, in the instance that I was showing, we didn't have LC resolution of the intact masses. We relied on the mass spectrometer to provide the mass resolution that we needed to pick apart the structural features. There are approaches now with different columns and methods that can be used on the LC portion to actually separate chromatographically the different DAR levels. And if you have that kind of chromatographic resolution, then you can look at those mass spectra independently. The other point there was that the fragments that are derived from the digestion, we often use a different kind of chromatography column. There's chromatography columns that are developed for intact mass protein analysis. So there's other ones that are developed for peptide analysis. So choice of column and gradients would be used to achieve the resolution that's needed for robust analysis.
All right, thank you for that, Brian. And for the sake of time, we're going to end the Q&A, but if there's any additional questions, we will reach out to each individual and address those. We thank you for your time and appreciate you joining us today.